Huh?
Let's do the male.
Let's do the male. Yes, we can. Okay. I'll try to do this fast. Male.
One last question about the pancreas.
Yeah.
You can't distinguish between the alpha and the beta and the
No,
delta.
not in an H&E you can't.
Okay.
I mean, if you specific immunostains are available to do those would be very expensive slides. It'd be nice to have that. Okay, male testis. There we go. Seminiferous tubules, right? So there you have a seminiferous tubule. There you have a bunch of
Where?
Leydig cells.
Yeah, like.
Leydig cells or interstitial cells. And LH, there's two bunches of them here. LH stimulates them to make testosterone, which diffuses in very high concentrations. And so now around the periphery here, these very darkly stained cells are...
Spermatocytes.
No, that's spermatogonia. The cells that you will see tend to be toward the periphery. Look at this one in particular. This, you see this? Give me something to point. Get a pencil out. Here's a really good example. You see this very irregular looking cell? That's a Sertoli cell nucleus right there. They tend to be sort of egg-shaped or irregular and lightly stained. So that's a Sertoli cell nucleus. And there's one. They're actually not. It's just the nucleus of the cell. Also, the more lightly stained ones, there also are some spermatogonia which are not heavily stained, but they're lightly stained, but they have very round-looking regular nuclei. These irregular ones, or elongated nuclei with this sort of variegated staining, those are Sertoli cell nuclei. But the heavily stained cells near the periphery are always spermatogonia. And the ones that look like these thin stripes of purple, those are spermatozoa, right, mature sperm. And then, because the spermatic wave is variously going on in different sections of the tubules, we look at one next door and we can see, for example, that there are a lot of spermatogonia and primary spermatocytes. Spermatogonia are small and they have some staining material. But to me, the most obvious cell, the largest cell in the seminiferous tubule is actually a primary spermatocyte. See how big they are. Not right near the periphery, usually inward in a little bit. You have some primary. This one, by the way, has a really good bunch of them. There's a lot of primary spermatocytes in that one.
So spermatogonia is only right beside the periphery.
What's that?
Spermatogonia is only...
The spermatogonium has this very dark but very dense, tight little nucleus. Whereas the difference between that and a primary spermatocyte is you can see the chromosomes. It's a large cell where you can see the chromosomes, that's the primary. Secondary is kind of hard to differentiate from primary. You have to sort of see them in context, and you usually don't see them together. So I wouldn't really worry too much about a secondary spermatocyte. It's really spermatogonia, Sertoli cell nucleus, a primary spermatocyte, and mature sperm.
No spermatids.
And spermatids, yeah. Yeah, and spermatids. Those are the little ones.
Tiny.
Yeah.
The ones right before the spermatozoa.
Yeah, so these would be good. Yeah, there's a spermatozoa along with spermatids.
You have to admit, these
You
are
don't see
hard
any
to-
primaries. What's that?
These are hard to differentiate.
Well, some of them are and some of them aren't. That's why I say spermatogonia, this is pretty obviously different from this, don't you think? These are primary spermatocytes, these are spermatogonia. Right?
Can you just give us one question on this and that's it?
You haven't even asked the question yet, and you're already bargaining. That's the most effective time to do it,
Just
right?
give us late Just give us late egg cells. Those are good.
Let's stick with Leydig cells?
Yes.
Well, I might. I don't know. Let's see. Anyway, Leydig cells are obviously there. So LH makes testosterone. It's FSH that stimulates the Sertoli cells
To do what?
support spermatic growth. And these, I'm not even going to ask you what these. You see these little round things with no nuclear staining, no purple stain. These are residual bodies. It's like eggshells. Sperm sort of hatch from eggs. Remember? Anyway, this is a very nice view of the seminiferous tubular testis. So I hope the slide that I get for the practical is that good quality. Are you still taking pictures?
You can save the slides that you show us for
What's that?
practical.
No,
They're
because
very
then you
good.
have to cover the labels and stuff. So Neil has them already made, you know. So, and I'll find out the day of whether any of them are what I want or not. But he's not going to be around Monday anyway. Okay, where are we going with this? Now we have prostate, seminal vesicle, epididymis, all of those things, right? Here's an epididymis. Epididymis, the key characteristic before you even see it, do you remember?
Sperm.
Sperm, yes. And there they are. Boom. So I may show you, this would show up pretty well on high power in a regular microscope, or maybe a little easier to show it here. You know, even though sperm are very small, that's pretty obviously the epididymis, the lumens. This is really like, the epididymis is really like a continuous tube that's very highly convoluted and ends up straightening out into the vas deferens. But you can see they're all filled with this stringy stuff. That's sperm. Okay, so that should be pretty obvious, the epididymis. There's nothing else that looks like that. And if you think about seminal vesicle and prostate, which look similar to that in a way, they shouldn't have any sperm in them unless you're having trouble, or the guy is having trouble. The sperm is getting backed up into those areas. So the only one that's going to have sperm in there are going to be epididymis. I suppose it's conceivable that you might have the oviduct with lots of sperm in there also, but these slides don't seem to have a lot of that. It depends, you know, these are, they could have taken it from animals right after they had intercourse. They could have had a lot of sperm in the oviducts. The oviducts are pure. So that's male. A lot of testis slides. Let's see, this looks like a prostate. No, it's a seminal vesicle. Okay. All right, key difference between seminal vesicle and prostate. You may remember. Way off. Okay, seminal vesicle. No sperm, so we know that is not epididymis, right? And we also know that it's not prostate because although you have a lot of these glandular lumens here in the seminal vesicle, there is very little tissue between them, right? Whereas in the prostate, you have a lot of stromal tissue, mainly smooth muscle. Here you have just a little. And no sperm. So in seminal vesicle, lots of glandular sections here with lumens, but the lumens are empty, except for maybe a little residual dry fluid or something, but no sperm, and very, very little muscle in between them, very little space. Whereas the epididymis has loaded with sperm. So that's how you recognize those two. Got a picture? And the way that you tell that different from the prostate is that the prostate has a lot of tissue in between. That's the stromal one. And also that was, remember there was an error in the book. This is this corpora amylacea, right? But remember, prostate, you have relatively smaller glandular sections here, lumens, and a lot of stromal tissue. The book was implying that that benign prostatic hyperplasia was a result of overgrowth of epithelial tissue. It isn't. It's this, that growing problem is this growing. When the epithelium overgrows, that's where the cancers arise, the so-called adenomas and adenocarcinoma is the typical prostatic cancer. That's epithelial, not stromal. Stromal is what you get from pH. So there is no correlation, just to let you know, in case you know, fathers, uncles, grandfathers, if they're having these growing problems, you don't have to worry necessarily about prostate cancer. There's no relationship between the benign and the cancers. It's not even the same kind of tissue. Yes?
What type of question would you ask about?
What type of question would I ask? I don't know. Do you have to how many times do you have to get up at night? Oh, you mean the test? Oh. Differently. Just thought I'd inject a little humor. What type of question? Well, the first kind of question I would ask for any of these things would be to name the organ or the gland. So to recognize, first of all, that it's prostate. The second thing would be to recognize what hormone it may secrete or what hormone it's a target. Now, this is not an endocrine gland. It's a target tissue. So what's it mainly a target for? If you have that growing problem, that's a commercial for a drug that does what?
Oh, castration.
Finasteride, yes. Which finasteride or dutasteride now, actually, which inhibits conversion of testosterone to DHT. So the active hormone is DHT. So it's a target for DHT.
Is that the same in the seminal vesicles? Or?
No. Remember those? You asked the question a little while ago about internal versus external genitalia. The prostate is grouped with the external genitalia because it's DHT dependent. The seminal vesicles, the epididymis, are the internal ones that are dependent on testosterone. That's a good question. So as far as so that actually gives you a good example of like B questions for seminal vesicles. Yes.
Any structures?
Any structures? Well, there's that corpora amylacea, which is really just prostatic fluid residue, but they give it a fancy name. So you have these corpora amylacea here.
What's actually inside the...
Yeah, that's right.
Okay.
Oh, I have to give you all back your lab reports too. Oh, we got time to do
None of that's stromal tissue.
Huh?
Stromal tissue.
That's all stromal
Both
tissue.
in this one and in the seminal vesicle. No.
Seminal vesicle. The seminal vesicle has very, very little stromal tissue.
Oh, well, okay.
That's the difference there. Excuse me a second.